So it's my great pleasure to be introducing our second Women in Cell Biology Early Career Award, following on very nicely from what Yuri was saying. And our winner this year is Lidia Vasilieva. Lidia has worked on RNA throughout her career. I'm just going to introduce her very briefly so that she can get on and tell us about her work. She was a graduate student in Finland, went and did her postdoc with Steve Wartowski at Harvard Medical School in Boston, and a few years ago now she became a Career Development Award. She was awarded a Career Development Award from the Wellcome Trust to start up her own lab in the Department of Biochemistry in Oxford. I'm very pleased to say that we learned about a year ago that she went through this big leap of making it from your first grant to your second, and she's now a Senior Research Fellow from the Wellcome Trust. And she's going to tell us today about her work, and just come up to collect her award. Lidia, congratulations. Thank you very much. It's quite a heavy story.
Am I supposed to open it? Oh, wow.
Compare the BSCB and BSCB
Thank you very much. It's a beautiful medal and really a great honor, I think. I don't think that all the credit should really go to me. I think this is really an achievement that should be credited to all the lab members and also all the collaborators that I have had and the great mentors that I met throughout my past. I think this medal is really special because I think it shows what a great progress we have made as a society in encouraging women, female scientists, to achieve, to pursue their goals and ambitions and reach their full potential. I think it's really, really important. And as Yuri pointed out, things really changed compared to just a few decades ago. So I think it's very good. Right. I need to switch to the. Oh, excellent. It's not beginning. Right. So back to science. I'm gonna tell you about the research that we do, which is focused on understanding the mechanism. Which is focused on understanding how cells regulate levels of individual messenger RNAs, which is a very important and understudied area of gene regulation. So now, as we all know, genetic information, all the living organisms has this amazing ability to pass on their genetic information encoded in DNA from generation to generation. And the way how information is organized within the DNA is in these individual blocks called genes. Each gene encodes for the protein that has, and each protein has a unique function in the cell. Now, on its own, DNA is actually quite inert. It's just a carrier of information. And what happens that the information from the DNA is transcribed to produce messenger RNA. Messenger RNA undergoes extensive processing reaction before it becomes functional molecules. And one of the important processing reaction includes removal of these non-coding parts of the precursor mRNA called introns. And it involves function of an important machinery called spliceosome. Now, RNA also gets modified at each end. It acquires cap structure and poly-A tail at the 3' end. Then it goes to cytoplasm, where it assembles together with the components of translation machinery in order to produce proteins. Now, each protein has a very unique function in a cell, and therefore it is very important for the cell function to regulate levels of individual proteins in the cell and make sure that the right amount of each protein is produced at the right time. Now, how do cells do that? Right, how slides look. In the way how it was thought that the cells regulate levels of individual proteins, until recently, the common view in the field believed that levels of individual proteins are mainly regulated through regulating transcription of the corresponding gene, and also at the level of protein synthesis and stability. What we came to realize now, based on recent work, including our work, that degradation of the corresponding messenger RNA contributes greatly to regulation of the levels of the proteins it encodes. And while we know quite transcription mechanisms involved in transcriptional regulation of individual genes has been studied for several decades, we know very little about mechanisms involved in regulation of RNA degradation in eukaryotes. However, what we do know is that this multi-subunit complex called RNA exosome is very important for regulating RNA stability in both nuclear compartment and also in the cytoplasmic compartment. Initially, this highly conserved complex was discovered in David Tollervey lab, and now we know that it really plays a very important role in regulating multiple cellular RNA species. And this slide shows a very beautiful structure from two labs, from Chris Lima lab and at Sloan Kettering, and also from Elena Conti lab. What we know from the structural work that these 9 subunits that on their own actually they don't have any enzymatic activity. They are assembled in a donut-like structure with the channel inside where RNA enters from the top in order to reach the catalytic subunit D3 located here in the bottom shown in purple. And D3 has 3' to 5' exa and also endonucleolytic activity. In addition to D3, exosome has also another exonuclease associated, shown in red here, associated with the primarily nuclear form of the exosome. Right. When activity or function of the exosome complex is deregulated in the cells, bad things happen. Mutation in the exosome subunits were found in multiple diseases. That includes blood cancers, such as multiple myelomas and leukemias, where catalytic subunit of the exosome was found to be mutated. Also, a very severe neurodegenerative disorders, such as spinal muscular atrophy, or SMA, where mutation in the subunits RP43 result in increased levels of myelin protein and multiple developmental defects of neuron system due to loss of function of the motor neurons. Another known disease is pontocerebellar hypoplasia, where OPSH, where RP40 subunit was found to be mutated, and the patient suffering with this syndrome is shown here. Typically, those patients are characterized with the overall muscular dystrophy, microcephaly, and also overall developmental delays. Now, given the fundamental role of this complex in regulating RNA stability in the cell, it's very important to understand how this machine, which on its own cannot distinguish one RNA from another, is in the cellular environment, is able to find its substrates. And this is the question that we would like to understand in my lab. In order to tackle this problem, we use simple organisms as a nice model system. We studied this problem in both fission and budding yeast. And what we have found is that exosome relies on proteins that we termed exosome specificity factors, or ESFs. And those proteins are important to recruit the exosome to RNAs that are to be degraded. And typically, this mechanism, well, there are multiple mechanisms, but typically ESFs also require RNA helicases. And so far, several ESFs have been identified, mainly in yeast. What is also interesting is that in contrast to the commonly accepted view that degradation is mainly limited to this, takes place mainly in the cytoplasm, we found that ESFs are actually recruited to the target RNA already co-transcriptionally, therefore kind of reprogramming those transcripts for degradation in the nucleus. And we think that degradation in the nucleus can regulate levels of the messenger RNA in the cell, and represents also a very important mechanism that is involved in regulation protein level. Today I'm going to tell you about one of these ESFs that is called MMI, that was found by several labs, initially by Yamamoto lab in Japan, to be important for degradation of meiotic transcripts in mitotically dividing cells. So if MMI is mutated, cells die, and the reason for that is that mitotic cells start accumulating meiotic transcripts, and that initiates meiotic program, which is really detrimental for haploid yeast cells. The reason why we became interested in MMI is that we found that in addition to previously identified meiotic RNAs targeted by MMI, we found that MMI can also target regulatory non-coding RNAs for degradation by the exosome. And that led us to speculate that its function may not be only limited to degradation of exclusively meiotic transcripts. MMI is actually quite conserved protein. It has this so-called YTH domain, which is also present in many other proteins in different species. Humans have 5 of these proteins. However, and those proteins have various functions in different aspects of RNA metabolism, ranging from splicing to regulating translation. They also found in high eukaryotes to bind to methylated RNA, which potentially can provide a possible mechanism for regulating recruitment of those proteins in high eukaryotes. Now, MMI is actually different, despite some resemblance on the level of the that you can see from this alignment. It actually has quite structural studies reveal that it has quite different binding pockets, and it interacts with RNA differently. In order to characterize the targets, MMI targets in the cell, we did this experiment called CRAC, which was developed by our collaborator, Sander Graneman. His lab is located in Edinburgh. So what you do, you cross-link your all the RNA binding proteins using UV light to RNA, and then you purify the protein of interest using very stringent conditions, so that you avoid pulling down RNA through protein-protein interactions, and only isolate RNA directly bound by your protein of interest. You also do partial RNA degradation in order to be able to map the binding site. And then you sequence cDNAs generated from this purification. And what we found is, we were quite excited about it, that in addition to RNAs known to be previously regulated by MMI, we pulled down 18 out of those, which is about half of those that were known to be regulated. The other half, I think, is probably not directly bound by MMI and were upregulated due to, as a result of changed expression of this initial regulators of meiosis. We pulled down quite a lot of about 400, well, to be more precise, 391 different protein coding RNAs together with MMI. This is the motif that we could bioinformatically predict that MMI binds. It's quite A-rich, and I must say it's actually quite frequently present on RNAs, so it's definitely not the only determinant of MMI binding, but it definitely does prefer this sequence. And this is one example of how the data looks like for this MAE4 RNA that encodes for the master regulator. It's a transcription factor, master regulator of meiosis. And this peak corresponds to the number of reads from cDNA that we generated from MMI purification. And these stripes here, they correspond to the presence of this motif. And you can see we can nicely align these binding events with the presence of those motifs on this RNA. I must say that in this particular case, there are multiple elements. However, on some transcripts, we observe only one element, and that's sufficient to recruit MMI. So the number doesn't really matter. And this is gene expression analysis, as you can see, compared to the wild type, where this transcript is entirely eliminated by the exosome. In MMI mutants, we see dramatic accumulation of this transcript. And consistently with the role of MMI global role of MMI in regulating RNAs, stability of those transcripts that are bound to this protein, when we normalize enrichment in MMI pull down for the abundance of the transcripts in the cell, we can definitely enrich for the RNAs that are present at a very low abundance in the cell, despite of the fact that they are very unabundant and underrepresented compared to other RNA species. What we were quite curious to find is that MMI binds within introns on some of the transcripts. And actually, for example, this one that encodes for ribosomal protein RPS15A, this is just a strange POMBI abbreviation of this gene. This one is really, really highly enriched in MMI pull down. So another transcript that where we saw very exclusive binding of this protein within the intron encodes for the essential helicase DBP2. It's an important player in RNA metabolism. It regulates splicing and also translation. It's present in both nucleus and the cytoplasm, and it's also highly conserved. The human homologs are DDX5 and 17. You might have heard about these proteins. They are known to be regulated during oncogenic transformation and also during development. Now, the fact that MMI1 binds these RNAs within intron allows us to speculate that MMI and exosome may be involved in degradation of these unspliced transcripts. And in order to test this, we decided to see if the levels of unspliced RNA derived from this gene DBP2 would be upregulated upon mutation of MMI or this non-essential subunit of the exosome called RP6 compared to the wild type. And this is northern blot, so you can see that as we predicted, we see upregulation of these unspliced transcripts. But what we were surprised to find is that also levels of the spliced RNAs appear to be even more upregulated compared to unspliced RNA, which was a little bit surprising because MMI exclusively binds to the intron, but in fact it actually regulates spliced RNA much more noticeably than the unspliced RNA. And this upregulation can also be seen from this RNA-seq data, where we see increase in the intronic reads and also in exonic reads. However, the nature of RNA-seq data is such that you can't really tell where these reads come from the spliced RNA or from unspliced RNA. Northern blot is much more informative in that respect. Now, in order to explain this, we came up with this model. So what we think is happening is that MMI binding to intron triggers degradation of the unspliced RNA. And that has an impact on how many RNAs get spliced. So what is really interesting here is that if we mutate the MMI and prevent these RNAs from being degraded, this unspliced RNA actually retains the splicing potential to be spliced while it stays in the nucleus. And this is the principal difference between degrading unspliced RNA in the nucleus versus degrading unspliced RNA in the cytoplasm by the mechanism like nonsense-mediated decay, where this is more of a quality control just to get rid of their unspliced transcript that can potentially generate toxic proteins. Here you actually, in addition to getting rid of potentially toxic transcript, you also regulate the levels of their final functional messenger RNA. So it also has a regulatory role, which is really interesting, I think. Now, in order to test this hypothesis, we asked, the next question we asked is whether intron is really important for MMI binding and subsequent degradation by the exosome. And in order to test this, we deleted this intron here that obviously prevented MMI binding to this transcript. And as a result, we see much more RNA being made. So all we do here, we don't change transcription, we just remove intron. So presumably it shouldn't really impact levels of the RNA, but it does, which is very similar to what we see here when we mutate exosome or MMI. Now, just to summarize, normally in the wild-type cells, binding of MMI to intron leads to downregulation of DBP2 mRNA. While if we prevent MMI binding and also splicing by removing this intron here, we observe accumulation of DBP2 mRNA. Similarly, if we delete MMI binding site, we also observe accumulation of DBP2 mRNA. So now we can conclude that it's actually binding of MMI to introns, which leads to downregulation of the levels of messenger RNA. Now, a very important question here is whether this pathway actually is used by the cell to regulate protein level, and under what conditions this is important. And in order to study this, we decided to do this assay looking at the ability of cells to grow under different conditions. And the way we do this in microbiology, we do different dilutions, serial dilutions of yeast cell, and we do spot them. And as you can see, that the mutants, if you delete intron, it actually under normal condition, ability of cells to grow is not compromised at all. They grow exactly as wild type. However, if we add high concentration of salt, which is a big stress for the cell, and you can see that even wild type cells don't actually grow so well. The mutated cells grow much worse, suggesting that presence of the intron and ability of cells to regulate DVP2 levels through the intron and MMI binding, which is lacking here in this strain. And you can see that in the wild type cells, upon addition of this high concentration of sodium chloride, cells downregulate the levels of DVP2. However, in mutated cells, they lack this ability, and we think that this is an explanation for this growth phenotype that we see. And we can see a similar effect also on the protein level. In the wild type cell, under these conditions, there is a dramatic reduction in the levels of protein. For some reason, cells need to regulate levels of this RNA helicase. We don't know why. However, in MMI mutants, we don't see this downregulation under this condition, very similar to what we see in the strain without intron. Now, just to kind of summarize this part of the talk, based on this data, we proposed a model where inefficient splicing can trigger degradation of the unspliced RNA through proteins like MMI. There may be other ESFs involved as well in a very specific manner. The RNA that undergoes splicing successfully goes to the cytoplasm and is being translated to make a protein. Now, under stress conditions, this balance between these two pathways, non-productive and productive, can be changed, resulting, let's say, in this particular case that I just described, in more RNA undergoing degradation and less protein being made. Now, we wanted to understand whether it's a splicing, regulation of the efficiency of splicing that is really a key factor to regulate this system. And in order to do this, we took advantage of our knowledge here that this splicing inhibitor, the reason why the splicing of this gene mRNA is inefficient is because of the splicing inhibitor that binds here. And we deleted this inhibitor, and what we expect to see loss of MMI binding, and as a result, increase in the levels of RNA. And interestingly, when we analyzed MMI recruitment to this intronic gene using chromatin immunoprecipitation, or CHIP, this is a technique that allows you to analyze the levels of protein recruited to chromatin. So you cross-link your protein to chromatin, and then you isolate your protein. You share your DNA, you isolate your protein, and you analyze the RNA, DNA associated with your protein using PCR, and these are different amplicons that we looked across this gene. And as you can see in wild-type cells, when splicing is slow, MMI is nicely recruited here. However, if we make splicing to be much faster, then we lose MMI binding here. And as a result, we see higher levels of spliced RNA being made. Now, this is just a summary of kind of take-home message of my talk. No? Still have time? Oh, great. What we think is happening is that this system is really regulated by ability of cells to fine-tune the efficiency of splicing, and that regulates the amount of messenger RNAs being made. By speeding up the splicing, cells can make more messenger RNA, and by slowing down the splicing, they can make less mRNA. Now, is this mechanism only a phenomenon? Is only unique for these particular cases that I described? I think that it's actually quite a widespread phenomenon. What recent studies demonstrated, and this is several recent studies in cerevisia by Dominika Libri lab and also in human cells, that at least in cerevisia, it's a quite a large portion, about 50% of intronic genes undergo very inefficient splicing. And you can see it when you mutate components of the subunits of the exosome, then cells start accumulating both spliced and unspliced RNA. So what I think is actually an investment in regulatory potential rather than just a waste. And under specific conditions, cells can regulate and change the balance. What we showed here is that the nature of this nuclear decay is such that RNA degradation is tightly linked to RNA processing. And this is really interesting because it can really regulate, fine-tune the balance. Once the pre-mRNA goes to the cytoplasm, it reaches the point of no return. What can happen to it, it can only be degraded. But while it stays in the nucleus, it retains the potential to be spliced. And what I really like to think that the levels, that the recruitment of ESFs could also be regulated. And that could lead to stabilization of those unspliced transcripts and generate more spliced RNA. Even so, we don't have the case for this now, but this is something that I would like to investigate in the future. And the reason why I think this, that the recruitment of ESFs must be regulated is that, as I mentioned previously, MMI sequences that we found to be really preferred by MMI, they are actually very abundant and very frequently present on multiple messenger RNAs, not only on intronic sequences, but they are not, in many cases, not bound by MMI, suggesting that something prevents MMI from being recruited to those transcripts, and we don't know, currently we don't know what. It could be a secondary structure, could be RNA modifications, something that was shown for other YTH domain proteins in high eukaryotes, could be protein-protein interactions, could be post-translational modifications, we don't know. But this is an avenue that we would like to go in order to understand this very first step, an important step that leads to degradation of RNA in eukaryotic cells. We also would like to dissect the order of events that downstream of the ESF recruitment that leads to degradation of these transcripts by the exosome. And finally, I would like to thank all the lab and most of all Cornelia Kilchek, whose work I presented today. She's a postdoc in the lab, and here Cornelia is on the hiking trip to the Lake District. And also, I would like to thank the wonderful collaborator, Sander Graneman, and our lab was funded by the Wellcome Trust. And Cornelia's salary was funded by German and Swiss foundations, DFG and SNF. And thank you very much for the attention. Please ask questions.
Congratulations for the medal.
Oh, thank you very much.
So now we have 10 minutes for questions. And,
so I finished early.
yeah, and that's good. Yeah, so we have a nice discussion.
That is something that would be really interesting to look
repeat
at.
the question?
Sorry. The question was whether binding of MMI changes under different conditions. I think it's a great question. We haven't looked at it, but this is something that definitely would be interesting to look. Yes? Uh-huh. I'm just hoping that I'm shouting. Right, the question was whether we know anything about the signaling pathway that talks to MMI. No, unfortunately we don't, but I think it's a great question. It would be really interesting to understand more about this.
Thank you.
Yes? Right. Yes, it's a little bit, I think, actually, sorry, keep. No, this is very good, thank you. So the question was whether, how do we control for how do we measure rate of splicing? This is a very good question, and I think this is, it's very difficult to actually measure it because of the degradation. In the wild type cells, because you really have to take into consideration, given this, the data I showed today, you have to take into consideration that the unspliced RNA, even if splicing is inefficient, it actually gets degraded. So if you just do RT-PCR on your total RNA, looking at the ratio between unspliced and spliced RNA, you may not actually observe any accumulation of unspliced RNA simply because it's unstable. So the way to look at it would be to do so-called net-seq, that looking at the nascent RNA when RNA is still attached to RNA pol 2. And nowadays, several labs have done this. Nick Proudfoot and Carla Nigebauer has published a nice paper looking at the efficiency of splicing in fission yeast, budding yeast, and I don't believe Nick really looked at the efficiency of splicing quantitatively, but at least in yeast it has been done, and I think this is really the way to go. And also, net-seq allows you to evaluate the efficiency of splicing in the wild-type cells, because you can never trust that there is no pleiotropic effect if you look in the exosome mutants. But this is something that we kind of try to do our net-seq version to evaluate the efficiency of splicing independently of using mutants. We did chromatin pull-down and looked at the RNA associated with the chromatin in order to be able to really evaluate, distinguish, kind of separate effect of the degradation from the effect on the splicing efficiency. Jordan, no? Okay. Yes.
Hi, so you mentioned other ESFs. I was wondering how similar they are to MMI and if the mechanism is also similar and what's
The
known about
mechanisms are very different. So there are only a few of those known, and mainly in cerevisia, in yeast. One is NERD1 that regulates non-coding transcripts and also some messenger RNAs. So the common feature between those proteins is that they are able to recognize RNA in specific manner. It can be RNA sequence only. It can be, well, obviously RNA sequence doesn't really explain the specificity entirely. There must be other factors. For example, Saccharomyces cerevisiae protein NERD1, it is able to recognize specific elements within RNA because it simultaneously binds to phosphorylated form of RNA pol 2. And this allows it to recognize these sequences in an appropriate transcriptional context. So it's more complicated than this. About MMI, we don't know what other factors or features within RNA or other protein-protein interactions that contributes to the specificity. But what we know is that the RNA sequence is just simply not enough. It is a part of it, probably, but it's not sufficient. Otherwise, the ESFs, they're quite different from each other, and the mechanisms are different, but the principle seems to be conserved. And all the known ESFs, they associate also with the RNA helicase. and the functional consequences, functional connection to the exosome is there, but the mechanisms are different. Jordan, yes?
So you showed that the MMI bound to many RNAs that are present at a very low level. And I was wondering whether do you think RNAs that are meant to be there at a low level, this will be how they do it, or is it actually those RNAs they're really actually trying to get rid of them, that there shouldn't be any there at all?
There shouldn't be, well, some of them, there shouldn't be any of those. So those meiotic transcripts that we pull down, they shouldn't be there. And you can see in the wild types, you literally, when you do RNA-seq to analyze levels of individual transcripts, you just don't see those meiotic transcripts at all. And then they increased, 1,000-folds in their abundance in MMI mutants. So it depends really on the RNA. For example, those intronic genes, they are regulated in the context of splicing. So obviously, it's not a black and white situation because there is a factor, the contribution coming from the splicing as well. So it really depends on the transcripts. No more questions? Okay, thank you.